Okay, so the composition of the blood, there are two really main components of the blood. One is plasma, and plasma makes up pretty good portion of the blood. But plasma is about 92% water. And then the other parts of the plasma are what we call blood colloid proteins. And basically that just means that they're like suspended in the blood. So they don't sink. There's another part of the blood called a formed component, and those things, when we like put them in a tube, they sink. And these blood colloid proteins, there's like three proteins that we talk about. One is albumin, and albumin is a big one because it carries bilirubin, which is a waste product in blood. But that makes about 60% of blood colloid proteins. And then there's fibrinogen. I can't spell. Fibrinogen is important because it's a clotting factor. It's I believe it's clotting factor 1A. We just learned that today, so I'm pretty sure it's clotting factor 1A. And then there's globulin. And that's just like other globular proteins, basically, in the blood. And those are all colloid proteins. They're going to stay suspended and they don't cross through the membrane of capillaries. So, like when we're talking about capillary pressure and movement of solutes and water especially, then through the membrane, the blood colloid proteins are what make up the BCOP, the blood colloid osmotic pressure. Along with plasma, we have formed components. I don't know that that's necessarily like the scientific exact term, but that's what I'm gonna call it. And that's made up of red blood cells. They make up about 45% of blood volume. And that obviously can change person to person. It can change due to elevation. It can change if you're like really sick and you're throwing up a bunch of water or you're like having diarrhea. Like all of those things can change your total blood volume, but your red blood cell count really isn't going to change. If you're at a higher elevation, you'll probably increase your red blood cell count so you can carry more oxygen because there's less oxygen available in the air. So we need a higher ability to carry oxygen. So we increase our red blood cell count, which increases hemoglobin. If somebody has something like anemia, that actually decreases their red blood cell count, decreases hemoglobin. So the big thing with red blood cells is they make up a good portion of our blood volume. For men, it's a little bit higher. For women, it's a little bit lower, and they contain hemoglobin. And hemoglobin is how we carry oxygen. That is how our bodies transport oxygen to and from tissues and the lungs through our blood and red blood cells. Another part of formed components is platelets, and these are also called cell fragments. Basically, it's a big cell that has fragmented into little parts, and we call them platelets. Platelets are also very, very important for clotting, which we're going to talk about in just a minute, but they're like absolutely key to clotting and the platelet plug. Without the platelet plug, we really couldn't clot. So that's really important. And then the last part is white blood cells. So all of your like macrophages, neutrophils, um, all of those things are going to be in the white blood cell layer. The platelets and white blood cells make up what we call the buffy layer. So if you were to put all of your blood, just a sample of blood, into a test tube and centrifuge it, you would get on the bottom, this would be red blood cells. And then you'd get a little buffy layer right here, and that would be your platelets and your white blood cells. And then you'd have plasma. And that's obviously not drawn to scale, but I'm trying. So The red blood cells, the percentage of red blood cells in your blood is called your hematocrit. So the things that I was saying that could increase or decrease, like the percentage of your blood volume that is red blood cells, that will increase or decrease your hematocrit. So like super high training performing athletes usually have a little bit of a higher hematocrit because they are demanding more oxygen to their tissues. So they have to be able to transport oxygen more quickly, so they have a higher red blood cell count as compared to like their total blood volume. Whereas somebody who's like anemic, so they have typically we talk about this in like women. A lot of women have anemia. They have low iron. That would be a low red blood cell count, and there's less iron being transported, which also you have less oxygen being transported because hemoglobin is a complex that transports oxygen and it contains iron. So that's kind of a tangent, but that's there. Um, we remove red blood cells from the circulation by breaking them down, obviously, but the big part that's kind of hard to break down is hemoglobin. So we can break down the cells, like the actual red blood cells. We just send them to macrophages and we say eat these. But then hemoglobin is kind of hard. So to break down hemoglobin, there's kind of two main parts. Hemoglobin is a complex, so it's made up of proteins, and there are two proteins. There's alpha and beta proteins. There's two strands of each. So there's alpha and beta strands. Um, and these are just amino acid strands, right? They're just proteins. So we send those as well. We send them off and we break these down into their amino acid monomers. So we break them down into the amino acid monomers. And then we can reuse them. We can make more hemoglobin with them. We can use them for transcription translation. I guess it would just be translation because that's where we're making a protein, but we can use them for whatever we want. The hard part to break down is heme. Heme is a little bit different. It's like the iron containing complex, and it has, oh my eye, the ability to carry oxygen. So this one, we are going to take heme and we it has some complicated bonds. It's not soluble in water, which means it's not soluble in the blood because the blood is a lot of water. Like obviously the plasma is 92% water and our blood is a lot of plasma. So the heme is not soluble in blood. We can't really transport it in our blood and do anything with it without it being in a red blood cell. So we have to break it down. And we first break it down to something called biliverdin. We do that by reducing it. So we add some oxygens. So we do this using, I believe it's called heme oxidoreductase. Reductase. I'm like reaching back into memory that I didn't know I had. Okay. Yeah, so I believe it's called heme oxidoreductase. That's going to add oxygens. So it breaks a bond between two nitrogens on the hemoglobin. And or on the heme, it's not hemoglobin anymore, it's just heme, and it's going to add oxygen groups. This polarizes the molecule, so it's polar and it's soluble. So it exposes some polar bonds. It makes some polar bonds by adding oxygen and it's soluble. It also has the ability to hydrogen bond in a few places. So again, it's very soluble in blood and we can transport it through the blood. So now we can send biliverdin kind of wherever we want through the blood. This has a blue-green color. And just like kind of an interesting fact is a lot of animals do not break down biliverdin any further than this. We do. We break it into something called bilirubin. But some animals, like for example, a bird or like a chicken, we have chickens at my house, so I just think of chicken. They don't break biliverdin down any further, and they excrete biliverdin out the same hole that they excrete their eggs out of. So when they are laying an egg, that's what gives the eggshell the bluish color that some chickens lay eggs because they're excreting biliverdin, which is a breakdown of heme. It's a byproduct of heme. Um, we then can break down biliverdin even further. We break a double bond, so we also are going to reduce biliverdin into something called bilirubin. And bilirubin has a yellow color. And because of the way that bilirubin molecularly is, it we're breaking a double bond that makes it so it can swivel. So we can swivel along those single bonds just because that's how chemistry works. Um, and so we can swivel along like these sp3 hybridized carbons, basically, is what's happening. And we're swiveling, and what happens is we get into a conformation where the bilirubin is interacting with itself polar wise, like its own polarity, and it becomes nonpolar again. So it's insoluble. So we no longer can transport it in the blood without a carrier, which we have, of course. So albumin, which we have a ton of in our plasma, is going to carry bilirubin for us through our blood and into our liver. And once bilirubin is in our liver, let me just draw my liver. That's a terrible drawing, but you get the point. We have bilirubin in our liver, and we add glucuronic acid. And the thing that happens when glucuronic acid is added to bilirubin is all of a sudden it's polar again because we're adding these big polar acid groups, right? Like we're adding this sugar that's really polar. Um, so it's like, oh, okay, well, I'm polar again, and now I'm soluble. So it becomes soluble again, and we send it down the bile duct because it's not going to be problematic solubility wise. And it does some really cool things in the intestine. Hopefully my drawing fits. I don't study on my iPad, so. In the intestine, we have now we call it conjugated bilirubin with this glucuronic acid attached. And it's going to help us emulsify fat. And in that process, it gets broken down, and those really polar groups get like ripped off because there are some strong interactions that happen. So we rip the glucuronic acid polar groups off, and we end up with oh my goodness, urobilinogen. And urobilinogen, again, is nonpolar again, so it can move in and out of membranes, whereas conjugated bilirubin cannot, unless we are like killing a cell or we force it out of the membrane through a protein. So urobilinogen can be further broken down. If it travels down the intestine, we can further break it down. So if it travels down into like our colon area, we have really cool bacteria in our colon that break it down into something called stercobilin, I believe. Saying it right, spelling it right, all the things. But stercobilin has a brown color. And this is what we excrete in our feces. So that's how we get rid of some of our bilirubin. Then there's like several other ways that we do it. So another way is this urobilinogen is sent out of the intestine, so it's no longer in the intestine anymore, and we send it into the bloodstream. And it travels through the bloodstream. It can go back into the kidneys and become conjugated bilirubin again. And this process, I believe, is hemopoiesis. I want to say it is, but I'm forgetting now. Or this urobilinogen can go to the kidneys. And in the kidneys, we break it down into urobilin. And urobilin has a yellow color, and it is excreted in the urine. So that's the other way that we can get rid and like fully excrete bilirubin, which is the end waste product of heme. So to get fully rid of heme, we have to go through this whole process, including our liver, our small intestine. Um, it can go through our colon or through our kidneys. But if there's a blockage in any of these places, then we would have a buildup of bilirubin somewhere along the way. Any of these derivatives of bilirubin, right? Because obviously we're breaking it down. Um, And a buildup of bilirubin, which is yellow, and most of these byproducts, except for stercobilin, which is brown, which is like what makes our feces brown. Um, Most of these byproducts are also yellow. So people that are having a buildup of bilirubin in whatever form it may be coming have like a yellow color to like their sclera or even their skin if it gets really severe, and that's called jaundice. And the way that we fix jaundice, especially in like infants who don't have the ability to add this glucuronic acid yet, their liver isn't fully formed and matured, so they don't have this glucuronic acid. They also don't have the gut bacteria to make stercobilin, so they won't have um, stercobilin being excreted yet. But these people, the way that we fix this normally is we treat it with a blue light, and blue light helps us to break a bond in bilirubin that makes it significantly more polar. So now bilirubin is soluble, just like conjugated bilirubin is, and we can send it so many more places when it is soluble versus insoluble. So that's kind of how jaundice happens and how we can fix jaundice. Okay, so there are three mechanisms that contribute to blood clotting. Um, the first one is vasoconstriction. So what happens is because you're feeling pain in that area, the pain receptors cause a response where your smooth muscle around that capillary or your arterial is going to contract, and there's blood flow to that region is limited. So blood flow limited to the region. And that's like if you think about, you give yourself a cut or like something like that. Obviously, we don't want to just be like gushing blood out of that while we're trying to create a platelet plug and clot it. So we limit the blood flow to that region by vasoconstricting. Then we have a platelet plug that's made, and we call that platelet aggregation. And this one's a little bit more complicated than just like one simple phrase. So I'm going to draw, you can capillary, arterial, whatever you want to call it again. Doesn't really matter. And it's lined with endothelial cells, which are just like specialized epithelial cells, basically that line cavities. And there's a membrane along this, we call it the basement membrane, and it's made up of a bunch of proteins, but the main protein in this membrane is, oh my gosh, collagen. So the main protein is collagen, which is important to remember for our clotting cascade. And let's say your blood pressure, we're inside this, and your blood pressure like absolutely skyrockets. And when our blood pressure is really high and we're pushing through blood through a capillary with a higher velocity, we kind of like tear off some of those endothelial cells, just like strip them basically off of the capillary, and we expose the basement membrane. We expose the um, sorry, collagen, and this causes a whole bunch of things to happen. So the first thing that is like important to note is that we have these platelets that are always floating around in our blood. They're just there. They're inactive right now, so we call them inert. They're just chilling, waiting for something to happen, right? We also have it's called the the VWF, which is just a factor that helps to activate platelets. So what happens is we have a receptor, the VWF receptor on platelets. So our VWF binds to platelets. And VWF also has a high affinity for collagen. So when VWF binds to collagen and to a platelet, it activates the platelet. And it makes it look kind of interesting, like a weird shaped octopus. Um, so it has all like these like little tentacle looking thingies, just like spurting out instead of being circular like it was before. And inside the platelet, when we have this activation occur, we have granules, and these granules there's two different kinds of granules in a platelet. There's alpha granules and then there's dense granules. And alpha granules, which I'm going to say are green, alpha granules release more VWF, which then obviously are going to activate more platelets, which are going to stick to the collagen. Well, the VWF is going to stick to platelets and be attracted to collagen. They're going to stick to collagen. They're going to be active, and then this thing kind of just keeps happening, right? So we have this amplifying effect when we start releasing alpha granules. It also releases PDCF, maybe PCDF, platelet driven cell platelet PCGF. P PCGF. I'm okay. Now I'm like losing the acronym, but basically, it's a growth factor that's released by platelets. So for some reason, the acronym has left me, but we release this by the alpha granule. And what happens is it makes it so the other cells are going to like multiply faster. So cells around it are going to be getting ready to heal and really increase the rate of the healing process because we're releasing this growth factor that I can't remember the acronym for. Um, then the dense granule, also, this releases factor 8. I think that's how you word it. Okay. The dense granule releases ADP and calcium. And those are like the important ones to remember. And then this ADP that we release from the dense granule is going to come and bind to platelets as well. And ADP also activates platelets. There's an interesting relationship that we have between ADP and prostacyclin because they both bind to the same receptor. Prostacyclin actually is an antagonist to that receptor. ADP is an agonist, right? So prostacyclin, which is a lipid, decreases coagulation. Um, ADP, which is going to activate platelets, is going to increase coagulation. It's going to help us create a blood clot. That's just like an interesting side note. Another thing that is released by, this is actually released by the platelet itself. The platelet membrane releases, it's called TXA2, which is thromboxane, which is just a prostaglandin. And it also can activate platelets. So there's a thromboxane receptor that activates platelets. So the main thing to get out of this is just that there's 3 activation pathways for platelets. And um, they all can kind of activate each other. And then once platelets are activated, there's a receptor that's expressed on platelets that is not expressed on inactivated platelets. And it's called the fibrin receptor. It has a fancy name. Um, it's like GDP two, 2B2A or something like that. Um, But we call it the fibrin receptor, and basically what happens is fibrin binds to the fibrin receptor. Fibrin is one of those important clotting factors that I talked about when we were talking about blood components, and then the fibrin creates a bridge between these platelets. So there's a bunch of fibrin receptors, and this is going to create a bridge between the platelets anytime there's a fibrin receptor. So we start creating this bridge, this like interwoven fibrin platelet glob basically that's going to plug that hole. We call it the platelet plug. So to kind of just recap, what happens is we're going to have a tear somehow in that membrane, and we're going to expose collagen. That exposed collagen, VWF binds to that collagen, and a platelet will bind to VWF. That receptor responds to mechanical stimulation. So if it's mechanically stimulated, so like if BWF is bound to collagen and then the blood is still rushing through and the platelet's kind of moving, but it's bound, then obviously that's going to open and create an activation of the platelet. Then that platelet becomes activated and releases things. releases substances from the alpha granule and the dense granule. The alpha granule, we're going to release more VWF, which is really important, and we're going to release a growth factor, which is going to help with healing. The dense granule, we release ADP, which is going to also bind to platelets, activating them. And then we activate those platelets, and that binds to collagen. And activated platelets are also going to be making thromboxane. Thromboxane is released from platelet membranes, and thromboxane also can activate more platelets. So we're getting three different pathways of activating platelets that then are building upon each other, because each time a platelet is activated, we're able to start that process all over again, and we're making more VWF, we're making more ADP, we're making more thromboxane, all those things. Activated platelets have fibrin receptors, and fibrin receptors are what's going to allow us to bind fibrin and create that bridge for our platelet. The third step in blood clotting is the clotting cascade, the coagulation cascade, actually, I think is what it's called. I don't know why in my brain it's called the clotting cascade, but this one is actually fairly complicated because there are three different pathways that we have to know. One is the intrinsic pathway, one is the extrinsic pathway, and then there's a common pathway. So I'm just going to start with the intrinsic pathway, and I'll probably take it through the common pathway, and then I'll do the extrinsic pathway and kind of explain where that ties in. But this kind of gets complicated because there's like a ton of factors that go into it, but I'm just going to try to make it as uncomplicated as possible. So if we have this collagen still in the cell, and just imagine that all of this is taking place right here where this platelet is, it just is like a little bit chaotic over there. So I'm just gonna ignore it. So we have factor 12, which honestly, I don't ever know if I'm writing these right. So if I am writing them wrong, then just ignore that. Factor 12, which is also called the Hagman factor. Um, that is going to bind to collagen. Collagen's going to activate it, basically. It's an enzyme. So these negative charges on collagen, it gets along with them really well. Collagen splits it, and it becomes 12A, which is activated. 12A is going to activate. Now that I'm thinking about it, this is not 12. 12 is next. Sorry, guys. 12A. There we go. It's going to activate 11 and make it 11A. 11A is going to activate 9, factor 9. And that makes it factor 9A. Factor 9A plus factor 8, they like to be friends. Factor 8 is a cofactor for factor 9, um, which just means that factor 9 needs it to work. Factor 9 then activates factor 10, and makes it factor 10A, which also needs a cofactor called factor 5. All of these factors are nameless to me. I have no idea what their names are. Factor 10 then activates factor 2 and makes it factor 2A. And factor 2 activates factor 1 and makes it 1A. That's a 2. Sorry about that. Makes it 1A, which works in conjunction with factor 13. They're friends as well. Factor 1 is named fibrinogen. It's like the inactive form of fibrin. Factor 1A is fibrin, which makes sense because obviously the end of our clotting cascade should end in fibrin, since that's what we're using to like basically create the platelet plug, and we're using it to bind things together. Factor 2 is prothrombin, prothrombin, and factor 2A is just thrombin. So thrombin activates fibrinogen into fibrin. Factor 10 activates prothrombin into thrombin, all that. All of this upstream of factor 10, okay, too far, is the intrinsic pathway. It's all things that happen inside the cell. So the big thing to remember there is the name of factor 12, which is the Hagman factor, and then just the order like of the factors, and then the names of these factors that are named. So this part is the common pathway from factor 10 down because this is shared between the intrinsic and the extrinsic pathways. The extrinsic pathway, we start with a cell actually on the outside of the membrane. So if we have the membrane of the Hurt cell, right, and we have the the basement membrane, and then we have a tissue cell or several tissue cells, they create this factor called factor 3. And factor 3 we also call tissue factor, probably because it's made by tissues. Um, and factor 3 also can be embedded in the basement membrane. Factor 3 activates factor 7 and makes it 7A. Factor 7A activates factor 10 and makes it 10A, and then we also need 5. So that's where this is going to join the common pathway, right? Um, Factor 7 also has a cofactor. It is vitamin K. So vitamin K helps to activate factor 7. So if we have a deficiency in vitamin K or we're blocking the recycling of vitamin K, we're going to be blocking the use of factor 7. Um, the other thing that's important is, or make sure, yeah, is breaking down fibrin. So when we break down fibrin, a couple things happen. Um, so when we need to break down the clot, we have to break down fibrin. I can't spell because I was thinking about clotting. Okay. So to break down fibrin, we have this thing called TPA, and it's actually um, tissue plasmin activator is what the name is, and it goes in with urokinase to plasminogen and makes plasmin. And plasmin takes fibrin and it chops it up into degradation products, whatever that may be. Um, so TPA increases the rate at which we break down fibrin because our bodies would break down fibrin anyways. Fibrin automatically is just going to kind of like dissociate. But by using plasmin, by creating plasmin and using plasmin to break down fibrin, we can break down the clots faster. This is important in like a clinical setting where maybe somebody has having a stroke or having a heart attack or anything like that. If we catch symptoms early enough, like within 90 minutes, if we administer TPA, then we actually can hopefully catch the clot fast enough and break it down so that those symptoms can be minimized and the effects of the heart attack or the stroke can also be minimized, which is kind of an interesting way to be treating that. Um, plasminogen is also just kind of chilling. It's not like anything crazy. It's synthesized in our bodies and just has to be activated. So we activate it using TPA and urokinase to make plasmin. And then we can break down fibrin faster. So then we go through that process where we're breaking down fibrin significantly faster than if we were to just let it happen on its own, like letting a scab heal on its own. Obviously, we're letting the fibrin break down and we're not using extreme amounts of TPA, like we're just using whatever our body has. Um, the other thing about the clotting cascade is there are ways to test the intrinsic versus extrinsic pathway. This is the extrinsic pathway, by the way, because it happens outside the cell, so we call it extrinsic, you know? Okay, yeah, feels really smart. There are ways to test it. There's a test called the PTT test. The PTT test tests the intrinsic pathway. It tests your Hegman, anything that has to do with like the Hegman factor. So it's going to test your ability to plot using the intrinsic pathway. So if you're on a drug like um heparin, then which heparin blocks your intrinsic pathway. Um, you would obviously have a harder time clotting, and that test is gonna you're gonna reveal that on a test. To test our extrinsic pathway, we use we use a PT test. Um, Now I'm wondering if I got that mixed up. But we use a PT test, which tests if you're using a drug like, oh, I just forgot the name. It starts with a C. Clotting? No. Coumadin? Yeah. If you're using a drug like Coumadin, which blocks the extrinsic pathway, it blocks factor 7 from working. Um, we can test that and see if we're blocking enough, if we need to be upping the dose, lowering the dose. But if we mix things up, then obviously gonna not work very well. Also, if we want to be altering pathways, we have to block a pathway from up high, because if we're blocking, like, say, factor 9, we're already 2 steps into the pathway, and there's like a billion factor 11s already. I realize that I'm drawing factor 12A again. That's a factor 11A. Right here, it should be 11A. Just go with me here. Um, there's already like a ton of them, so if we are putting a drug in that's supposed to be blocking that, then I mean, we're not really going to be effectively blocking. But if we're blocking like factor 12, which is at the very top, like we're blocking the Hagman factor, then that's a big deal. If we're trying to block like prothrombin, which is at the very bottom, and you're coming from two pathways to make prothrombin, like you're screwed. That's not happening. Like you're not going to block anything that's coming from there. Like if we're blocking prothrombin or thrombin or fibrinogen or fibrin, like that's probably not going to work, because we're so far down in the pathway that we're making like billions of these things. Like that's really not going to be an effective way to treat anything. So that's just a thought.